what is peptide and polypeptide? Peptide is the stretch of amino acid where the number of amino acid ranges from 2 to 50 amino acid, and the amino acid are bonded together by covalent linkage of a peptide bond called as the amide bond. When two amino acids are bonded together, then they form a stretch of peptide or stretch of amino acid called as the peptide. But the number of amino acid residues must be less than 50, then it is called as peptide. But when the number of amino acid residues in a stretch of amino acid is more than 50, then they are called as polypeptide. Means the peptides, they are in higher number. When two amino acids come together, then the carboxyl group of first amino acid and the amino group of second amino acid form an amide bond by the removal of one water molecule. This bond of formation between carboxyl group and amino group is called as amide bond. This is the figure showing the stretch of a peptide, small peptide, where each and every peptide or a polypeptide has left side as N terminal, where the first amino acid residue have free amino group, while the right side of that peptide or a polypeptide always has a free carboxyl end, or called as the C terminal end at the last amino acid residue. When a stretch of polypeptide or a peptide is seen clearly, then you can see a number of amide bonds between the successive amino acid and forming the stretch of peptide or a polypeptide. The average molecular weight of an amino acid is 128 Dalton. But in a polypeptide or a peptide molecule, the average molecular weight of an amino acid is 128 minus the molecular weight of water that has been removed between the amide bond formation, and then the average molecular weight of amino acid in a polypeptide will be 128 minus 18, that is 110 Dalton. The length of the peptide bond is 1.33 angstrom, that is in between the bond length of carbon and nitrogen single bond, that is 1.49 angstrom, and carbon and nitrogen double bond, that is 1.27 angstrom. That is the average bond length between the amide bond, and that is equal to 1.33 angstrom. Now a question arises in our mind that what is a peptide bond? Peptide bond is also called as a amide bond. That is a bond formed between two amino acid, and when two amino acid come together, the first amino acid have a carboxylene and the second amino acid has a amine group. The two ends of the two amino acid come together and a water molecule is removed, forming an amide bond. In a peptide or a polypeptide, when a number of amino acids come together, they form a number of amide bonds and form a stretch of peptide or polypeptide. The carboxyl group of first amino acid and the amine group of second amino acid come together and a water molecule is removed, that is from the OH of the carboxyl group and hydrogen of the amine group. When this is removed, the carboxyl group of first amino acid and the amine group of second amino acid forms a bond that is called as the peptide bond, having a length of 1.33 angstrom, also called as the amide bond. When a number of amide bonds are formed between different amino acids together, they form a stretch of peptide or a polypeptide. Now one more property of the peptide bond. When between the two amino acid peptide bond is formed, then immediately they are arranged in a trans configuration, where the alpha carbon of one amino acid, this is the alpha carbon of one amino acid, and this is the second amino acid, and the amide bond is formed between this carboxyl group of first and the amine group of the second. Then immediately they are arranged in a trans configuration in the opposite direction, and the torsion angle between them is 180 degree. This trans configuration makes amino acid more stable, and most of the proteins of our body are in this configuration. And the most important point to remember is that the torsion angle between them, means they are in the opposite direction, that is 180 degree. While the second configuration of the proteins or the peptide bond is the cis configuration, where the alpha carbon of one amino acid and the second amino acid alpha carbon are in the same configuration or on the same side, their molecules are on the same side, and the torsion angle between them is 0 degree, and it makes a peptide bond less stable, and so most of the protein immediately form the trans configuration for their stability for a longer time. Now, what is Ramachandran plot? Ramachandran plot was given by G.N. Ramachandran. He has mentioned 4 to 5 points, where the first point was that the bond between carboxyl group and alpha carbon is a pure single bond. Second point, that the bond between amine group and the alpha carbon is also a pure single bond. Then the third point, he has mentioned that the torsion angle between carboxyl group and alpha carbon is called as psi, and the torsion angle between amine group and the alpha carbon is called as phi. He has mentioned the permitted value of this phi and psi in a peptide bond, and he has also given the non-permitted value where the peptide bond is not going to be stable. The permitted and non-permitted value all were given by G.N. Ramachandran. When the permitted value of this phi and psi are together plotted on a 2D plot, then that plot is called as Ramachandran plot, and it shows the permitted value of phi and psi where the peptide bond will be stable, and it makes a protein to persist for a longer time. Now, coming to the protein structure. The basic important information about a protein structure is that protein is a structure that doesn't have a branching. It's an unbranched structure where it's all made up of only 22 amino acid. It has 4 basic structure, and these are primary, secondary, tertiary and quaternary. The simplest form of a protein is the primary protein structure, where amino acids are serially arranged and they form amide bond between them and forming a stretch of a protein. The primary protein structure is the basic building block of secondary, tertiary and quaternary protein structures. So, we will start with the primary amino acid. Coming to the primary protein structure, that is the simplest protein structure, and it is initially formed and its base of secondary, quaternary and tertiary structure. The primary protein structure is formed by the linear arrangement of amino acid, where the amide bond or the peptide bond is formed between them. The first amino acid is bonded to the second amino acid, then to the third, and the peptide bonds are serially formed between them, and they are serially arranged in a linear way, forming a primary structure of a protein. When this primary structure is formed, further they form a secondary structure like beta pleated or alpha helical structure, or tertiary structure, or the quaternary structure. And when they are folded in a different way or they form a helical structure, they form the basic structure of the protein of our body. Coming to the secondary structure of the protein. A protein can have a secondary structure. It may be alpha helical structure or beta pleated sheet structure. Alpha helical structure is a rod shaped structure which forms a helix like structure. It can be right handed or it can be left handed. Most of the proteins of alpha helix are in right handed fashion, forming a helix, and this helix is stable because of the hydrogen bond formation of the oxygen of carbonyl group of one amino acid and the hydrogen of amine of the second amino acid present in the next turn. When a helical structure is formed, about 3.6 amino acids are present in a helical turn and its pitch length is 0.54 angstrom. When it forms a helical structure, the rise of the amino acids occur and it is about 1.5 angstrom. This helical structure is very important in our body and most of them are right handed. Coming to the beta pleated sheet structure, that is also a secondary structure of the protein. Here in beta pleated sheet structure, they form a sheet like structure where more than two polypeptides are arranged serially, and there the beta pleated sheet structure is also stable due to the hydrogen bond formation between the hydrogen of the amine group of one amino acid of one strand and the oxygen of the carbonyl group of the second amino acid of the second sheet. Similarly, when there are more than two sheets in a beta pleated sheet structure, then this type of hydrogen bond is formed between the second sheet and the third sheet and the third sheet to fourth sheet, and a beta pleated sheet structure is formed. The distance between the two amino acid in a beta pleated sheet structure in one strand is 3.5 angstrom and each strand is called as beta strand. This whole structure is stable by the hydrogen bond. Means the secondary structure of a protein, if it's a alpha helix or a beta pleated sheet structure, both of them are stable by the hydrogen bond formation between the oxygen of a carbonyl group of one amino acid and the hydrogen of the amine group of second amino acid, whereas in alpha helix it's in between the two amino acids of a helical turn, while in the beta pleated sheet structure it's between the amino acid and carbonyl group of one sheet to the another sheet. Now the third or the tertiary structure of a protein. The tertiary structure of a protein is a 3D structure. We know that in a 3D structure a molecule is present in the x, y and z direction, that is a 3D structure. In tertiary structure, a protein has a number of bonds, a number of polypeptides are here and they form a 3D structure having a number of forces that or the interaction that make it stable, and it include hydrophobic interaction, that is a non-covalent bond. Second, it include electrostatic interaction that include the salt bridges. Third, it include hydrogen bonding. Fourth, it include van der Waals forces, and fifth, it also includes a covalent bond, that is a disulfide bond. All these 5 forces or interaction make a protein stable in a quaternary structure, and that is forming the some basic structures of an organism. The last and the most important one is the quaternary structure of a protein. The quaternary structure of protein is made up of a number of polypeptides known as the subunit. When two subunits or more than two subunits are arranged together, they form a multi-protein structure, and the best example is the hemoglobin that has a number of subunits. The subunits of a multi-protein structure or a quaternary structure may be homo subunit, means they are similar in structure, they are same, or they may be different, or hetero subunits. This multiprotein structure or quaternary structure is made stable by two types of interaction, that is covalent and non-covalent. The covalent bond include the disulfide bond, while the non-covalent interaction or non-covalent bond include hydrophobic and electrostatic interaction. These three bonds or the interaction make a quaternary structure of a protein stable and useful for our body. The last point about the protein is that when a protein is synthesized and it works in our body, that may be a simple protein or a conjugated protein. A simple protein is a protein that is made up of only amino acid, and the best example is serum albumin. The simple protein is the basic structure and it's very important also for the working of our body. It only has the amino acid, but conjugated proteins are also important for our body, and they are very important. A conjugated protein is a protein that has a protein part and a non-protein component. When this type of protein is having a part of non-protein component or the conjugated part, then only that protein get activated. This type of protein is called as the holoprotein, and the protein part of that holoprotein is called apoprotein, and the non-protein component is called as the prosthetic group. When apoprotein and prosthetic group come together, they form a holoprotein, and then that protein get activated and that work in our body. The best example of a conjugated protein is glycoprotein, present over the covering of a cell. This glycoprotein has a carbohydrate moiety or carbohydrate component with the basic structure of the protein. The second is the lipoprotein that has a lipid molecule with the basic structure of a protein. Metalloprotein that has the metal ions with the basic structure of the protein, and the hemoprotein that has a heme group or the iron containing compound with the basic structure of protein. These four examples are conjugated protein, and they are also very important for the basic structure of our body, and they are very important. Now the protein or the amino acid topic is over, and in the next class we will study about carbohydrates.